We're going to take a look at the different types of light microscopes in this lecture. The first type of light microscope that we're going to start with are the bright field microscopes that you use in lab. So with all the microscopes we're going to talk about, the only things that you need to know are the things in red. So the big advantage of using these bright field microscopes as compared to the electron microscopes is that you're able to look at living organisms as long as they're not stained. So that's their big advantage. And then the disadvantage of the bright field microscope as compared to an electron microscope is that it has much lower resolution. And remember, resolution is just how sharp or clear the image is, how much detail that you can see. And so the resolution of the bright field microscope, you can see down to the size of a bacterial cell, but then anything smaller, it can't resolve that, and so anything smaller, you can't see that. The next type of light microscope is the dark field microscope. So how it got its name is that the background is dark and the image of the specimen is bright. So because the background is dark, that's how it got the name dark field microscope. So the big advantage of the dark field microscope and why it was developed is that you're able to look at living cells without having to stain them. So it really increases the contrast without staining. And one of the uses of the dark field microscope is that it's really good to view a type of bacteria. They're called spirochetes, which we'll learn about later. And one of the spirochete bacteria, these are called Treponema pallidum. And this is the cause of the disease syphilis, which is one of our sexually transmitted diseases. So here's a picture of Treponema pallidum bacteria looking through the dark field microscope. So notice that the background is dark, and then you can see the bacteria is very bright. So bright image with a dark background, that tells you it's a dark field microscope that took this image. And these are living bacteria. And if you click on this at home, this YouTube video, it actually shows you the Treponema pallidum bacteria viewed through the dark field microscope. And you can actually see them swimming around, so it's pretty cool. So definitely check that out at home. And these are spirochetes, so they're kind of a spiral shape, and so you can kind of see they're kind of a corkscrew shape there. And then this other picture on the right, these are volvox. This is a eukaryotic microorganism, and you can see that they are green. They are not stained, and these are actually algae. These happen to be multicellular, so there's a group of them that live together. And these were actually first discovered by Van Leeuwenhoek. So he was looking at pond water, and he's the first one that discovered the volvox algae. And these are not stained at all. And that's the big advantage of using these dark field microscopes, because you can look at these living microorganisms, see what they're doing when they're alive, and you don't have to stain them to try to increase their contrast, because the dark field microscope increases the contrast without having to stain the cells. You don't have to know this part at all, but if you're interested in how they create the image in the dark field microscope, this is how they do it. So they place a dark field stop, which is placed right above the light source. It's a piece of plastic that only lets the light through on the outer edges there and stops the light going through in the middle. And so the light that hits the specimen will go through the lens and then up into your eye, so that'll create the bright image of the specimen. But the light from the background doesn't go into the objective lens, and so that doesn't make it to your eye, and that's why the background appears dark. So that's where the name dark field microscope came from. The phase contrast microscope is the next type of light microscope, and this one is pretty different. The name says what's going to happen. So we're going to be using some differences in the refractive indices from the specimen versus the background, and that's going to have the light waves become out of phase with each other, and that's going to create contrast in the image, and that will actually increase the contrast without having to stain the cell. And so that'll allow us the advantage of being able to look at living cells without having to stain them. So the phase contrast microscope will increase the contrast without having to stain the cells. Because remember, if you stain the cells, then that ends up killing them, and then you can't see what they're doing when they're alive. So let's just take a look at a picture of images created by these phase contrast microscopes. So the first image on the left, this is an amoeba, one of our eukaryotic microorganisms. It's actually a protozoan. So it has these pseudopods or false feet that it puts out, and it crawls around, and then it ingests its food. So it's like an animal. And so this cell has not been stained at all. And so you can see, a lot of detail inside here. So we've really increased the contrast without having to stain the cell at all. And so we can look at it while it's still alive. And then the picture over on the right is another eukaryotic microorganism. This is a paramecium, another protozoan. And again, this has not been stained at all. And just by having the light waves out of phase, that has increased the contrast without having to stain. And again, you don't need to know this part at all. This is just how they get the light waves out of phase and create these images. So above the light source on the bottom of the microscope, they have an annular diaphragm. So it has a ring that the light can go through, and we'll hit the specimen, and we have part of the light waves that go through the specimen. They will end up being out of phase with the light waves that go through the background. And there's another plate up here that the light waves will go through. This is the diffraction plate or phase ring, and that will also help get the light waves out of phase even more, and so that will create the contrast to really make things stand out without having to stain the cells. And then this is just comparing the three types of light microscopes that we've talked about so far. So all of these pictures are looking at a paramecium, and we've got the bright field microscope on the left, the one you're used to working with in the lab. And then here's the dark field microscope, and then this is the phase contrast microscope that we just talked about. The next type of light microscope is the differential interference contrast microscope. This one is going to use some prisms to create two beams of polarized light that are at right angles to each other, and that will create an image that will create a lot of contrast, and that'll allow us the advantage of looking at a living cell without having to stain it, and that will really increase the contrast. So that'll be the advantage of using this differential interference contrast microscope. We can look at living cells. And it really increases the contrast without having to stain the cells, which is good, because if you stain the cells, then you kill them. And the other thing that's unique about the differential interference contrast microscope is that it gives us a three-dimensional image, which is really cool. So here's a picture generated by a differential interference contrast microscope. So this is of a paramecium, and you can see how the image really pops off the page here, and it really looks three-dimensional. And this is not stained at all, and you can see there's a lot of contrast here. You can really pick out some of the structures inside this paramecium here. And so we can look at this living paramecium, see what it's doing without having to stain it and kill it. So that would be the advantage of this differential interference contrast microscope. And again, you don't need to know this part at all, how it works, but how this microscope works is we've got a prism down here above the light source that will separate the light into two separate beams, and that will go through the specimen. And we've got another prism up here that will then combine the light sources together again. And because of the differences in the refractive indices of different parts of the specimen and the background, then that will create the contrast that we're going to see in the image. Okay, and then our next type of light microscope is a fluorescence microscope. And with this one, we're going to be staining the cells with some dyes that are called fluorochromes. These are fluorescent dyes that when you shine ultraviolet light on them, they will fluoresce. So it's really easy to recognize images that are made with a fluorescence microscope. So like you can see on this image on the bottom right here, you can see these really bright fluorescent colors, so you know that this was made by a fluorescence microscope. A lot of times we're going to use immunofluorescence to make our images. So what happens is we end up taking antibodies that we've attached the fluorescent dye to, and these antibodies are very specific to what they will bind to. So maybe we've made them so that they will bind to the nuclear envelope. And then maybe we've got different antibodies that maybe will bind to the microtubules inside the cell. And maybe we've got a different fluorescent dye for that. So maybe we've got blue for the nucleus, and then we've got the green for the microtubules, and that allows us to pick out the different parts that are inside the cell. So you get these really, really pretty pictures with that. But the disadvantage with these fluorescence microscopes is because we are staining the cells, that kills them. So the disadvantage of these is that you can't look at living cells. So they're all going to be dead. And that's because you've had to stain them. Okay, and you don't need to know this part at all, but it's just showing you that if you shine these fluorescent lights onto your specimen that has been dyed with these fluorochromes, the fluorescent dyes will then give off the fluorescent lights, and then that will make the image. image. And this is just showing you the immunofluorescence. Immuno just referring to the immune system and just talking about how we're using antibodies. So we've just attached to our specific antibody a fluorescent dye. So maybe we want to know if our patient has syphilis. So we're looking for the Treponema pallidum bacteria. And so we can have antibodies that will only bind to the Treponema pallidum bacteria. And we can take the sample from the patient, add in those fluorescent labeled antibodies, and then turn our ultraviolet light on and see if anything fluoresces. And if we see this under the microscope, then we know that our patient does have syphilis, and then we need to treat them with antibiotics. Okay, and our last type of light microscope is a confocal microscope. And this is just a special type of fluorescence microscope. So we are going to be staining the cells with our fluorescent dyes. So we'll use our fluorochromes. But we're going to be using a laser beam and a computer to make our image. And we end up making a three-dimensional image, which is really cool. So you can see that over here on the bottom right here. You can see how the image kind of pops out and looks three-dimensional here. So that would be from a confocal microscope. And you can also know that this is from a confocal microscope. So besides being a 3D image, you also see these fluorescent colors. So that's how you recognize that. And then the disadvantage of the confocal microscope is because you're staining the cells with the fluorescent dyes, you're killing them. So the disadvantage is you can't look at living cells. Okay, and you don't need to know this part, but how the confocal microscope works is we're going to be using a laser beam that will shine onto our specimen here, and it's going to focus on one plane of the specimen at a time, and then that laser beam will then be reflected up to our detector here, and anything that's not in that plane of focus won't be detected by the computer. And so we'll just focus on different planes, and then the computer will then take each plane of focus and then put all those together to make the three-dimensional image here that we're looking at. So this image here is really very interesting. What they've done is they've taken a red fluorescent dye that will stain dead bacteria red. And then bacteria that were still alive before you stain them, they will take up the green fluorescent dye. And what this is a picture of is a biofilm. And remember, biofilms are really hard to treat medically because they've got that slimy layer, and it's hard for antibiotics to penetrate, and also hard for our immune cells to get in there and get rid of those bacteria. So what they've done with this biofilm is they treated it with antibiotics, and now they're looking at it under the microscope to see, well, did we kill all the bacteria or not? And you can definitely tell that the antibiotics were not able to penetrate through that biofilm, and we've only killed off the surface bacteria of the biofilm. And you can see that the bacteria that were deeper down in the biofilm, they're still alive, or they were before we stained them. And we did not get rid of the bacteria in the biofilm. So that's why these biofilms are very hard to treat if you have a patient with one.